Thanks for the invitation to be a part of this. We've been engaged in a genomic selection program now for about 8 years with sugar kelp here in the northeastern U.S. Not nearly as long as Asian countries, but perhaps we may be the most advanced, at least in the Western world. I want to point out our colleagues at University of Connecticut, Charlie Yarish's lab, Dr. Jean-Luc Janik at Cornell University, and Sergei Nudzin's lab at the University of Southern California, and also our Common Garden farm at the University of New Hampshire, the steady farm we've used for the last 8 years. So I'm going to try to answer this question that we are all trying to answer. How can kelp breeding be conducted responsibly? First, just put this in the context of what's going on with kelp around the world. I think most of our audience understands that changes in kelp abundance and distribution are occurring at an unprecedented pace, fueled by warming waters and other anthropogenic effects like fishing down the food chain, leaving urchin barrens scouring kelp beds. But it's happening very unevenly across the world. Some species like Laminaria ochroleuca are expanding their range northward as they're more warm tolerant. Saccharella latissima sugar kelp is also perhaps increasing in abundance toward the poles, even though its southern regions are diminishing. And other species are facing local extinction with these kind of pressures. Very interesting modeling study here recently published showing that habitat loss without some sort of genetic intervention in the North Atlantic will, by the end of the century, under these three different climate scenarios, will reduce the habitat of these four important species by a third or a half. But if there were some genetic intervention, i.e. inducing thermal tolerance, increasing thermal tolerance of the kelp species, the models show that half of that loss could be recovered. So in terms of kelp restoration and habitat management, these interventions may become necessary to preserve our fisheries. And so our selective breeding program looked at ways to quickly and effectively employ temperature tolerance. We did this starting with a study of gametophytes, where we stress tested them up to 24 degrees Celsius. We did this in 96 well plates where we could measure their health and fluorescence. And we determined that we could find 3 pairs of putatively tolerant gametophytes and 4 pairs of putatively intolerant gametophytes, and we created sporophytes within those groups. And after 12 days of stress testing those crosses of sporophytes, we could see very clearly a difference between the tolerant strains on the left side here, the 4 pictures there. So these are the results of our 96 well stress test with gametophytes, tolerant pairs on the right, intolerant pairs on the left. We created sporophyte from within those intolerant and tolerant strains. These are results of the 12-day stress test showing the the tolerant ones on the left 4 pictures and the intolerant results on the other 4. I've got your, oops, sorry. There we go. You can still see my screen, right?
Yes, we can.
Okay. So this graph summarizes the data here, basically the total area versus the healthy area. And we could see that those that were sporophytes from the intolerant strains lost healthy tissue much faster than those from the tolerant strains. So this is just an illustration of ways we can quickly screen our gametophytes to potentially select for heat-tolerant sporophytes. Now, as all this climate catastrophe is happening in slow motion, I think the underlying principle here is that kelp farming is actually one of our possible climate solutions. And I don't want to lose that sight of that fact that that's what motivates many of us in the academia as well as the industry. And this WWF infographic shows very well all the variety of products that our society depends on that come from fossil fuels, but could alternatively be derived from kelp polysaccharides and other compounds in kelp. Um, so here's just a quick summary of our genomic selection over the last 7 years. We're currently in our 8th year, just harvesting that these last couple of weeks. Um, and it shows generally that, first of all, yield is highly heritable, and that after many random crosses in the first few years and the development of a genomic selection model, we can now target strains that are almost 3 times the average commercial harvest within the Gulf of Maine, which happens to be about 6 kilograms per meter. We've now generated about 40 crosses that are over 15 kilograms per meter. And these are detailed in the publications that I cite in the bottom there. I'm happy to send those to anybody who's interested. And along the way, we've developed public resources for breeding. This is not something that's going to be put on a shelf in an academic institution. We're actively collaborating with other academic and commercial operators. We've accumulated over 2,500 different gametophytes, males and females, isolated from sporophyte collections from 30 locations. That diverse collection is available in a public repository, the National Center for Marine Algae at Bigelow Labs. And furthermore, we've characterized much of this germplasm with gene sequencing, with the sporophyte traits that are associated with different pairs of gametophytes, and that's all cataloged in a public database, sugarkelpbase.org, for the community to use. And we've generated, based on our population genetic studies, peer-reviewed population genetic studies, we have divided our research and breeding programs into 2 areas, one for southern New England, south of this dotted line, and one for the Gulf of Maine above that dotted line. Oops, sorry, I'm going the wrong way. So a central question for the regulators and for the stakeholders all involved here on this call is, can farm strains impact the wild? And there are many different ways that farm strains could impact the wild. It doesn't take much imagination to go there. But one of the things I want to emphasize here is that we have attained some amazing plots, like this one, 28 kilograms per meter fresh weight. They're not Frankenkelp. They're derived purely from genes that were 1 generation removed from the wild, natural genes assembled from those localities. But I think the question also is, what does farming now, on a very limited scale, what possible impact can it have? And again, assuming these strains are selected regionally appropriately based on population genetic studies, there's a very low risk of interbreeding impact, particularly given how much wider, how much more numerous wild kelp are compared to the farm population. Now that's the case in our current day, but many people have ambitions for a much more expansive farming future. Large farms, kilometers wide and kilometers long. And I just highlighted how wild kelp in certain regions of the world are not doing so well. So all of this is very site specific. If you have a wild kelp population on the southern range of if its distribution, and it's not doing very well, then putting a farm right next to it and swamping it with potential genes that are different from the wild could be a big problem. So we've been developing parallel to this breeding effort. And a very important part of this breeding effort is to also develop non-reproductive kelp, or NRK as we call it for short, as a possible solution for working in parallel with selective breeding. And we've created dozens of crosses over the last 6 or 7 years. Many of them are relatively high yield as well. So we want to keep wild kelp wild. As kelp breeders, that's the responsible thing to do. That is the resource from which we need to go back to at some point. It's the natural gene banking that's occurring. Even though our anthropogenic effects are likely to upset those, we need to keep the wild wild. And so we've been focusing on three different ways of creating non-reproductive kelp. One is selecting for natural mutations, and we do this by bioinformatic detective work that prevents spore generation. And this is a patent pending process owned by the four academic institutions I mentioned in the start of my talk. And that patent and licensing is being negotiated with a startup company called Macrobreed. And full disclosure, I'm a co-founder of Macrobreed and an advisor. There are also two other ways that kelp could be, the non-reproductive kelp could be produced. And I think Bertrand alluded to one of them, hybridizing species that are non-reproductive, like mules. And a third way that the Chinese have published most recently, creating triploids, typically by the crossing of diploid by normal haploid parents. So I just wanted to outline one possible way for breeding progress. It's to match these two different paths I've described. And to take a page from our own Mariner program, we've been testing and selecting these locally but diverse reproductive crosses on a very small scale. We started with 1 meter plots, but as you'll see me standing on the right up here, pulling a half-meter plot out of a harvest tote, that's 10 kilograms, and that gets really hard on my back. So we've gone to about 1 meter crosses that have between 100 and 200 blades. That's plenty to phenotype across. Second thing we've been doing is developing and testing fail-safe, because we're working with the local populations, non-reproductive kelp, understanding that at some point our breeding program is going to diverge dramatically, and as we domesticate and create strains that are focused for local markets or specific markets like higher fucoidin or lower iodine. We are working with third-party partners to develop a certification program, because we know there's going to need to be an arm-length certification for this that's not part of the regulatory establishment or an academic lab. And we're also see a future where hopefully commercial expansion will be allowed for NRK once it's been well established and well certified. They could possibly expand beyond local regions. So we might have multi-regional strains at some point. And with that, I think I'll conclude and just acknowledge our funding sources. And I'm looking forward to the next discussion and generation of questions and answers.